Your body is a physical system constantly fighting the second law of thermodynamics. You burn roughly 70 watts continuously, spending energy to repair DNA and maintain voltage just to keep the cellular machinery running. If aging were solely governed by physics, the result would be a uniform, gradual wearing out. Your body would slowly erode into dust, and there would be no biological mechanism to intervene. Curious, but in 1961, a biologist named Leonard Hayflick, working at the Wistar Institute, observed something strange. Human cells appeared to have an internal limit to their lifespan. Hayflick cultured human cells in nutrient-rich environments, assuming they would divide indefinitely. Every single culture stopped dividing after exactly 40 to 60 replications. The cells were counting their own divisions and pulling the brake on schedule. This meant the cells weren't breaking down from random physical damage. They were following a precise set of instructions. Defining aging as a biological program reclassifies it as a software execution, a process that, in theory, can be edited or revised. To understand how cells count, we have to look inside the nucleus. This is an image of human chromosomes. Capping the ends of each one is a repeating sequence of non-coding DNA called a telomere. It acts as a protective buffer for the genetic data. Every time a cell divides, the molecular machinery that copies the DNA misses the very last section of the strand. This geometric limitation causes the telomere to shorten slightly with every replication until it hits a critical length and the cell stops dividing. The body possesses an enzyme called telomeres that rebuilds these protective caps, but in most adult tissue, your genome suppresses it to prevent cells from dividing endlessly, keeping cancer in check. In 2012, Harvard researchers reactivated this enzyme in elderly mice and observed systemic regeneration. Shrunken spleens and livers returned to normal size, and brain volume measurably increased. The biological clock is a two-way street. When the suppressed enzyme was restored, the underlying cellular hardware immediately resumed its work, proving it retained the ability to rebuild itself. The telomere counter is only one mechanism. Sitting on top of your DNA is a second system known as the epigenetic layer. This acts as the cellular software, dictating which specific genes are active and which are silenced. This system changes in such a predictable pattern that researchers at UCLA developed a method to calculate biological age by tracking the chemical methyl tags attached to your DNA. It's known as the epigenetic clock. If this software controls biological age, we can potentially reboot it. In 2020, Dr. David Sinclair and his lab took aged blind mice and introduced specific genes that reset the epigenetic state of their retinal cells. The mice regained their vision. The researchers hadn't altered the DNA sequence itself. The cells were behaving as if they were young, because the instructions telling them to act old had been overwritten. The Sinclair experiment proves that cells can retain their youthful function even after decades of decline, provided the epigenetic data wreckage is cleared. But what happens to the cells that reach their telomere limit and stop dividing? They don't always disappear. They persist in your tissue in a state called senescence, the zombie cell. These cells are actively toxic. A senescent cell secretes a continuous broadcast of inflammatory molecules, damaging the surrounding healthy cells and accelerating systemic aging. The background stiffness in your joints, or the lingering ache after exertion, represents the measurable output of this inflammatory broadcast, which has been accumulating in your tissue for years. A new class of drugs currently in human trials, called senolytics, targets this exact problem. They selectively identify and clear out these toxic cells while leaving the healthy tissue intact. Aging is an active degradation driven by our own cellular wreckage. Science is finally figuring out how to sweep the tissue clean. To understand why our cells are programmed this way, we have to look at evolutionary economics. The disposable soma theory points out that in a harsh environment, natural selection optimized our bodies for rapid reproduction, not long-term maintenance. There was zero evolutionary payoff for maintaining a human body past the age of 40. Surviving to 65 is an engineering achievement that wildly exceeds the original design specifications, written for a world of predators and famine. Nature already proves that biological decay is negotiable. The Greenland shark maintains its body for 4 centuries in the deep ocean. The Turritopsis dohrnii jellyfish can entirely revert to its juvenile state under stress. The programming inside us is an evolutionary compromise rather than a fixed constant of the universe. For the first time, we are attempting to renegotiate those terms. While pharmaceutical interventions navigate clinical trials, the programmable layers of aging are responsive to intervention now. Your epigenetic clock responds directly to resistance training, deep sleep, and stress management. You can support the physical energy requirements of cellular repair through mitochondrial support. Compounds like NAD+ precursors and CoQ10 supply the cellular turbines producing the energy necessary for maintenance. Specific eating patterns, like caloric restriction and time-restricted feeding, activate a process called autophagy. This forces your cells into a maintenance mode, where they actively digest and recycle their own damaged internal components. These habits act as specific chemical instructions, forcing the biological operating system to prioritize maintenance over growth. Lift, sleep, fast, repair. The physical decay of the universe is a thermodynamic law, and that cannot be stopped. But the biological programming running on top of it is a negotiation, and we are finally learning how to argue back.